Yes. Good evening, Pooja. Good evening, Pooja.
Good evening, sir.
How are you, Pooja?
I'm fine, sir. How are you?
Yeah, I'm doing good. I'm not able to take class last day because there was an important commitment, so I was not able to take class for you.
Okay.
Yeah. Okay.
Okay, sir.
So Pooja, we are focused on the integral pro. Okay, we are focused on the integral protein, how they help in transfer of molecules, and the role of. I think we discussed about the structure of plasma, the basic structure of plasma membrane. Then we got the elementary ideas
Yes,
about
sir.
the chemical composition of plasma membrane and how plasma membrane helps in the transport of proteins. And we are focused on the importance of channel protein or the passage protein or tunnel protein, or which is also called as transmembrane protein, right?
Yes, sir.
Any doubt in this, Pooja?
No, sir.
Okay, fine. So now next we're going to continue with the plasma membrane alone. So I told you we have an integral protein, it's also transmembrane protein, and told you about the amino acids how they are formed. So this also we have completed, and this also we have completed about the receptors and how they go with the enzyme functions. Now, the topic we have is in the organization, what are the other constituents apart from proteins? What are the other
Mm.
constituents which are there in the plasma membrane? So, plasma membrane not only they made up of lipids and proteins. Can you say how the organization of lipids is arranged in a plasma membrane? Yes.
Yes, sir.
Can you just tell me how it is arranged? Only lipid part
Sir,
alone.
we can see. Yeah, here we can see the blue layer, sir. The
Hmm.
blue layer head is called as glycerol, and now we can see the tail,
Hmm.
fatty acids. The purple, uh, purple, green, red, purple. The four things are integral proteins, sir.
Hmm.
And above we can see a green color, purple color, red color. Uh, a purple color, below purple color, green color, sir. There are peripheral proteins, sir. And the yellow thing is like the receptor, sir.
So yeah, so very good, beta. So not only this, apart from it, if a carbohydrate is joined with a protein, it will form a glycoprotein. Or to a lipid layer, if a carbohydrate is joined, they'll form as a glycolipid. So together they form an important component called the called as glycocalyx. So main thing is to give you the protection to adhere the two cells together and cell recognition like ABO blood grouping, which I'll be teaching you. For all this, we require these proteins. So these proteins one is going to act as a receptor, or they're going to act as an that is called cell recognition as a receptor, or they act like a protection or to join two different cells also like cohesion property. They require this following organization. So I told you lipids are amphiphilic in nature. What is amphiphilic? Amphi means what?
They have both properties of hydrophobic and hydrophilic.
Yes. So did you see? Receive the recording of the last class?
Yes, sir.
Yeah. So we have a dual purpose here. One is we have the head and we have the tail, which is actually a glycerol followed by the fatty acid, the long chain fatty acid. The head is completely hydro.
Hydrophilic.
Yes, they are water loving, hydrophilic. They focus outside. The tail end is focused inside. That is called as
Pic.
hydrophobic. Hydrophobic end. So we
Pic.
have a hydrophilic end and hydrophobic end, which gives them an amphiphilic nature. That is, they are made up of two loving property. One is water loving and one is water hating property. So this is how the properties is given. So this is the head and the tail. So we have the head which is focused towards the aqueous environment because they're water loving. Tail, they are focused on, sorry, this is the tail, the long chain fatty acid, which are hydrophobic and they are projecting towards opposite to each other and they kept away from the aqueous environment. Okay, beta?
Yes, sir.
So, general organization, this is what we're going to see here. So, the functional architecture, that is a lipid organization. Already we have discussed about that. Anyway, we'll see about that.
Sir.
See, you look about here, self-assembly in different structure. That is, you know what is micelles or mono or bilayers or liposomes. Anyway, I'm going to teach you that also. Have you learned about the cleansing action of soap?
Yes, sir. I learned it, sir, but I don't remember it,
Yep,
sir.
you don't remember, no problem. See, if you have here some oil dirt, which I'm giving you in black color.
Mm.
This is your oil dirt.
Okay.
So when you put some detergents
Okay.
and all, in detergent they'll form one organization called micelles. What does this micelles says that the tail, it's oil. So the tail area of the
Mm.
fatty acids are attached to the dirt.
Head.
And this is the head part.
Okay. Ah.
And if this is your bucket,
Mm-hmm.
in this bucket, this is the water. And this is the
Mm-hmm.
head part. Head part is water loving. This is the tail part. It's water hating property. And this is your cloth. To understand, this is your cloth. In the cloth, what will happen when we just dip it in the soap solution and take it up? So when you're going to pull it up, what will occur? This is the water loving hen. They will pull the head along with them. They also will be pulled, and dirt molecule also will be removed. So when you put some oil in the oil, if I add a detergent solution, the lipids will be arranged in this following format.
Okay.
So this is the water loving end, this is the water hating end. So this will get attracted towards the water. So they will form one tiny structure like this. This is called as micelles. Got it?
Okay, sir.
This micelle formation
Yes,
occurs
sir.
between the dust particle, or you can see that fat molecules with another fat molecules, they'll form a micelles. Bile will emulsify with the
Okay.
fat and they form micelles. Have you learned this term? Bile will emulsify the fat and they form fine globules. Globules will form micelles. The term இங்க கேள்விப்பட்டிருக்கீங்களா? இல்ல.
I don't know, sir.
Now, if we have a water molecules, listen here. So this is how the micelles are there. So in micelles, the head is projected outside, tail is projected inside. So wherever the water is
Yes,
there,
sir.
they are projected. So it's fully surrounded by
Mm-hmm.
water. If only one area is surrounded
Mm-hmm.
by water means imagine that they can form monolayer or a bilayer also. In monolayer, if only one particular portion, if you have the water, the head part will get attracted towards
Mm-hmm.
water and the water hitting end, that is called the fatty acid, they are
Uh-huh.
focused towards the air. At the same time, if fully both the areas when they're covered with water, they'll form one, two.
Uh-huh.
They'll form bilayer. Yes, ma?
Yes, sir.
Yeah. So they'll form bilayer when it's fully surrounded by water. Here you can see one, two. Layer number one, layer number two. At the same
Two.
time, if only one area is water, other area is air means they'll form a monolayer. So, since
Yes, sir.
liposome means when they are fully focused towards the water, two lipids are lipo means lipid, soma means body. So, they will form a lipid surrounding body that is called as liposome. Got it?
Yes, sir.
So, this is how the energy is there here. You can see here the lipid layer, water loving end is focused towards the water, water hitting end is focused towards the inner layer. You got it better?
Yes, sir.
But micelles, mono and bilayer. Mono layer between air and water. When they are fully surrounded by water, this lipid have an organization as a bilayer. When it's fully and largely surrounded by water, they form a lipid bodies that is called liposomes.
So liposome means many bodies of bilayer, lipid bilayer. We can take it as liposome if it is surrounded by water.
Yes, in a simple way, I can tell you. Listen here. Just split this word. Soma. What do I mean by soma? Chromosome, color bodies.
What
Soma means what? Body.
body?
What do you mean by lipo?
Lipids, fat.
Lipids. So it's a lipid body. Got it.
Yes, sir.
So we have lysosome, breaking body. Got it. So when you can see
Yes, sir.
liposomes, when you see some hormone like steroid hormones are there now. Steroids.
Mm-hmm.
Steroids have
Mm-hmm.
this following structures. It is actually made up of a bilipid layer only. But what happens? We
Uh-huh.
can lower the drugs. That's the main function. Lipo function they help in um, drug designing. They help in drug designing. We are again coronavirus or again new virus discovering something, right? All this are used in pharmaceutical purposes because they can easily load the drugs into the liposome and they can target to the concerned organ. Okay,
Sir.
got it.
Yes, sir.
So if you look about your it's a simple topic. Look about your functional architecture about the membrane symmetry. They are asymmetrical in distribution. Because I told you one point you can remember here. I'll show you here. The distribution of lipids is not always equal. In your liver, you are a human
Mm-hmm.
being. By liver cells, it is 52 percentage. Erythrocyte is 49 percentage. In your neural cell, it is 79 percentage. In your mitochondria, it is 24 percentage. That means what? The distribution of fat layer is not at all uniform. Got it?
Yes, sir.
So asymmetric. What do you mean by asymmetric?
Uh, they are not equal. The
Yeah.
sides are not equal.
They are not equally distributed. Okay, so asymmetric distribution of
Yes, sir.
lipids refers to an it is actually an uneven distribution of lipids across the cell. Uneven distribution of lipids across cell membrane, across different cells of a cell membrane. This is called as asymmetrical distribution of lipids. Why it is important means why it is varying அப்படின்னு கேட்டிங்கனாக்கா every cells have a different functions, right?
Yes, sir.
So we look about here, this is your exoplasm, this is your cytoplasm. We look about here, the distribution of lipids is not uniform. Why this is important means, for example, if I take here, okay, I just if I give you the points, you can understand this. If I put as PC, what is PC? Okay, sorry, what?
Phosphatidyl choline.
Okay, I'll just go with the first one. What is SM?
Sir?
What is SM? Spingo?
Sphingomyelin.
Very good. You're doing your biotechnology, right, beta?
Yes, sir.
MSc biotechnology or BSc biotechnology?
BSc Biotech. So here, like here, it is like 5 year program, sir. 2 years we spend it like a preparatory year, sir.
Hmm. So you're there in the preparative year.
No, I was supposed to go to this year, sir,
Hmm.
but due to my health issue, I was not able to. I went to school, and the school told me this year, due to my health, it is not suitable for me. So they told me to come next year, sir.
Okay. So SM stands for sphingomyelin. PC stands for phosphatidyl choline. Okay. What is PS?
Phosphatidyl serine, sir.
Very good. Phosphatidyl or phosphatidyl serine. Okay. So like this, we have all names and all, like we have CL and all these are actually nothing but a type of phospholipids. So these are the different
Mm-hmm.
types of phospholipids. Anyway, these names are enough for us, I think so. Even if I can go for PE alone now. What is PE?
Phosphatidyl ethanol amine.
Very good. So here you have an alcohol along with an amino acid.
Yes,
Correct.
sir.
So what is the main role is they help in, we're going to discuss about that new topic now, signal transduction,
Mm.
which is also called as cell signaling. These are there in your peripheral protein, not on the integral protein. Okay, beta. They also help in membrane fluidity and they help in cell receptor response to identify the cells. So the distribution of lipids should not be equal. It should not be symmetric. It should be asymmetric. At the same time, if there's any
Sir.
change in the composition of these lipids, it can lead to the cell death. So any change
Okay.
to the distribution of lipids leads to cell death. Why? Come on, try to answer. Why?
So because the composition is changing, no, sir. Because the cell is
The composition is different,
for
na?
cell.
I'm saying that if it is actually your
Uh,
your plasma membrane, though made up of 54% of proteins and rest of them are lipids, the lipid component that was
yes,
there in
sir.
your plasma membrane should not change for that particular cell
Yes, sir.
organelle. Why
Yes,
they
sir.
should not change? What will occur? What any change to the distribution of lipids leads to cell death? Why? Because a cell want to communicate between two different cell. The cells has to allow the entry and exit of molecules. The cells have to do their metabolism. For all this, the amount of lipids is fixed based on their biological function a cell will perform. Any change in the biological composition may result in metabolic disorder, which may lead to a new propagated cell or it may lead to cell death also. Got it?
Yes, sir. Yes, sir.
Now, a term I use called fluidity. What do you mean by fluidity?
Sir, I guess like a fluidity, we know. So the cytoplasm is filled with fluid.
Hmm.
I guess for the movement of molecules that has entered through the integral proteins, the cell that is needed, the molecules that needed by the cell to move through the cytoplasm.
Hmm. Okay, I'll show you one small experiment, okay, which they have done on the plasma membrane to know what is fluidity. Then you can
Okay.
understand the role of flipases or transverse exchanges. For this, one theory is there, okay, beta.
Okay.
So listen here, what the scientists have taken is I'll take a green color. I'll just take a cell alone. It is a mouse cell. To this mouse cell, they added an antibody called as anti-mouse green fluorescent. Anti-mouse green fluorescent antibody. It is actually a marker. Uh, in your college, did they taught you about antibody marking mechanism or trail mechanism?
Oh,
No,
sir.
no. Okay, it's a
No.
mechanism by which we see. They say that what is the source of oxygen in photosynthesis? Is it actually water or it is actually carbon dioxide? Which is the source of oxygen?
Uh,
If you forgot that, it's a 10th portions. It is actually water molecule will split, right? So you learned about the
okay.
water molecule will be splitting into the corresponding protons plus corresponding electrons plus a molecule of oxygen is liberated, right? So even carbon dioxide is
Okay.
entering into the cell, but they are turning into glucose only, not for evolving the oxygen. So how do they identify that even carbon dioxide? If I remove the carbon, they also can give me the oxygen. But how do they
Yes, sir.
identify that carbon helps in glucose synthesis? The oxygen is liberated
Mm-hmm.
only from the water means for this you have to use some markers, right? To water I have to give some color coding. To carbon I have to use a color coding technique. So that's called color coding technique for understanding. Okay, better.
What's color coding, sir?
Color coding is called as just you just make a note in your notebook. Biomarker identification, just put as biomarkers for biotechnology. A lot of knowledge we can gain, but anyway, but it's not there for you, but in the upcoming days I'll be teaching you. So now this is a biomarker, right?
Okay.
When I add this to the
Okay.
biomarker to the cell here, what happened to the cell means when I used fluorescent green, and when I observed the cell membrane was green in color. Only the cell membrane was green in color. Next, they took one human cell. To this, they added anti green, anti man, green, fluorescent antibody. So, what scientists have observed is,
Mm-hmm.
Pooja, this and all they will not teach you. Okay, they thought they think that you could have learned in your lower classes. But anyway, but you didn't learn
Okay.
it because 11th, 12th, you didn't study in our school, right?
Yes, sir.
If you are there, then I could have been your teacher.
No, sir. Actually, what happened is 11th, I went to another school,
Hmm.
and by 12th, I got COVID and my health spiraled, sir. So I went to NIOS, sir.
Okay, you went to NAOS. I didn't know. So where did you do your NAOS course?
It So I don't know the place. It was I it was like a play. I don't know. It
That's
was
fine.
somewhere in Coimbatore, sir.
Okay, no problem. Listen here. Now this is when I add anti-man red fluorescent antibody. It was completely coded by, that is, the plasma membrane was giving me only red color fluorescence. So they all were labeled by red color. Okay.
Yes, sir.
So what is actually fluorescent is it's actually a powder. It's a powder only. So when we add this powder, they mix with the plasma membrane and they gave you this color. Now what scientists have observed
Mm.
is, listen now, this is very easy to understand. Now they took a mouse cell. Now they took a human cell. To this, they fused these two cells. So it is called as fusion, as a two different nuclei are joined, right? So it is called as hetero. What do you mean by hetero? Others. Hetero means two different. Okay. Okay.
Yes, sir.
Carry-on means nucleus. So what do you mean by hetero carry-on?
Mm. Two or more nucleus.
Yeah, two nucleus are joined in a single cell. That's called heterokaryon. Okay, so in a cell, two different nucleus are joined now. Now to this, what they added is they added anti-mouse green, they added anti-human red. Both was added. So what they have observed is they observed that half of them were able to give you green, half of them they able to give you red. There is half of the cell, or the cell means when you look up at the plasma membrane, it was green and it was red. Then what they observed is when they heated, the triangle symbol, see it is heated. When they heated for 37 degree Celsius, what they observed is half of them were red and half of them were green. But the distribution was different. So when they observe with the different temperature, what they observed is when they heated at 37 degrees Celsius, the content of green and red fluorescence was equally mixed but in a alternate proportion. Now what happened? They kept the same cell, they kept it
Mhm.
0 degrees Celsius. When they kept at 0 degrees Celsius, there was no change. Again, when they heated for 37 degrees Celsius, they had some changes. Again, when they kept at 0 degrees Celsius, there was no change. So, why at a 0 degrees Celsius there was no change? That is the question. Why at 0? I'm asking you. Why at 0 degrees Celsius
Uh,
there is no change?
Sir, I guess at 0 degrees Celsius, I guess they have not, there was no supply of any heat energy, sir. And I guess at 37 degrees Celsius, they have applied some heat energy.
Okay,
So
to
that could
understand
have
that, I'll
caused
tell
us
you, you
the.
take some oil and keep it refrigerated, maybe coconut oil. What will happen?
Uh, uh,
It will solidify, right?
yeah,
So, what proteins they will not solidify, but what about lipids?
they solidify.
Can solidify. That is why when I kept in 0 degrees Celsius, there was no change. When I transferred to 7 degrees Celsius,
Okay,
there was some changes. Again, there's no change when I
okay,
brought into 0 degrees Celsius. There won't be any changes.
okay, sir.
This is because of the
Okay.
fluidity of the plasma membrane. So this says that plasma
Uh,
membrane is dynamic, dynamics always active, and the fluidity or the plasma membrane is because of lipids, because lipids generally solidify at the below 0 degrees Celsius. At lower temperature, lipids will solidify. So there was no much net molecular movement of antibody green and red, whereas in others there was,
mm.
they were able to see some movement. Are you able to understand this experiment?
Yes, sir.
Now you see this. What is this lateral diffusion? That is, these molecules are getting transferred. Can you see here?
The glycerol is getting transferred.
So
Yes,
the red
sir.
one is getting transferred to the next down, and this one is getting transferred to the next one. That is
This one.
between them or here. This is actually called as rotational movement of the lipids. Why they are occurring means because lipids, when they go exposed to higher temperature, this one how they got exchanged, this mechanism. They had red, only red, green, green.
Mm.
But when they had a equal ratio of red and green, that is when I added, one was coated with red, other was coated with green. So there was no problem.
Okay.
But when I heated them, green, red, they interchange the position, right? The lipids position have been interchanged the position. That property
Yes,
is
sir.
explained here, and this is catalyzed by an enzyme called as flipases is the name of the enzyme, which helps in shifting of the fat molecules or the lipid molecules present in the plasma membrane from one location to the another location. Okay.
So when we apply some when the temperature when we add some temperature other than 0 degrees Celsius with the help of enzyme flameases, there will be an interchanging of position of lipids, sir.
Yes, and this mechanism is called as transverse exchange mechanism occurring by means of enzyme called flipases. Don't say 0 degrees Celsius. Lower
Okay.
temperature, it will not be exposed. Higher temperature, the lipids will be exchanging their position, which helps them to bring about the transfer of molecules. Since that is the right term I'm going to use is the plasma membrane is dynamic in nature. Dynamic means what? Always they are active.
Active.
Got it?
At sir, at only at a higher temperature the exchange of
Yes, at lower
fat molecules
temperature, yeah.
take place, or
No, at the room temperature there was no much changes. At higher temperature only, that is in done in the laboratory called in vitro. So only at 37 degrees Celsius, the position of this fat molecule is interchanged.
okay. Sir, I'm sorry, sir. Is it at particularly at 37 degrees Celsius, or as it the temperature goes on, the interchanging will also happen?
Now, water maybe the change in temperature, your body temperature will not change because we all are warm-blooded animals or homeotherms, right?
Okay, okay, okay.
The
I understood,
homeotherm
sir.
temperature is 37 degrees Celsius. In 37 degrees Celsius, that is transverse exchange of molecules takes place. So plasma membrane is active. If
Mm.
transfer will not take place, means plasma membrane is not dynamic. They're not functioning. That means they may result in cell death.
Yes, sir.
Got it?
Yes, sir.
Please have a look on the diagram first. Any doubt you can ask me. Don't take more than 1 minute for
Mhm.
this.
Yes, sir.
Once done, please let me know.
Yes, sir. Done, sir.
Yeah. So are you able to understand this? What is
Yes,
this
sir.
lateral diffusion or transverse movement? How the fast movement of the molecule occurring is because of the lipid nature, not because of the protein nature. Okay.
Okay, sir.
So this is a model which was proposed by Singer and Nicholson in the year 1972. I think that maybe the date of birth of your maybe uncle or aunt, someone in your family, right?
Sir?
1970s means it may be the date of birth of your maybe uncle and aunt.
Ah, yes,
Yeah.
sir.
So that age, in the year 1972, a lot of models were there, beta. But there was a model called as in the year 1972, two different scientists. One is called Jonathan Singer, and second is Garth Nicholson. So it's called Singer and Nicholson. So they proposed, they didn't derive it. It's actually a proposed model. About the structure of plasma membrane, they proposed one diagram. So that diagram only enabled us to understand that the biomembrane is actually not solid. Why they should be solid? Lower temperature they become solid, but a higher temperature they are not at all solid. They are actually have a state called liquid state. That is, they are being viscous, and they have a phosphorus containing
Mhm.
lipid and a phosphorus containing proteins are also there. So the molecules are not arranged in a uniform manner. Because of that, what will happen? New proteins are formed. These proteins are exposed towards the environment.
Sir?
Yes, got it?
Yes, sir.
Yeah. Now, this is the fluid mosaic model of plasma membrane, and the name of the scientist is Singer and Nicholson. This you could have learned in your grade 11. If not, it's also fine. I believe we're going to see in depth. So what I discussed is
Yes,
a
sir.
fluid mosaical model of plasma membrane. So you have got some diagram here to know about the fluidity. So you know about what is saturated and unsaturated. Are you aware of saturated fatty acid and unsaturated fatty acids?
No, sir.
Okay. You take coconut oil, and coconut oil when you keep in the refrigerator, what will happen?
They solidify.
That is called as saturated fatty acid. Okay. At the same time, you take cooking oil and keep in the deep refrigerator also, will they solidify?
No.
You check. Okay. Just have a take half glass, maybe a small glass, half glass of refined. What type of cooking oil you use in your home? So when you just
Oh,
check,
should I do it now? Okay.
yes, yes, ma.
No, sir, I'm waiting for the explanation.
Yeah, yeah. So I just want you to just observe this diagram. So fluidity is because of the plasma membrane. The fluid is because of the presence of lipids in the plasma membrane, not because of the proteins. Proteins cannot give
Mhm.
you the fluidity. Only the lipid molecules
Mhm.
alone can give you the fluidity. Are you able to know understand?
Yes, sir.
So this fluid nature of plasma membrane is making them dynamic, which we have understood by the previous experiment. This experiment.
Yes, sir.
Now, this is actually your head. You know the structure, just a small recall. This is your alcohol, the head. The head is completely water.
Loving.
So they are called hydrophilic.
Philic.
This one is called as the tail.
Hydrophobic.
Cut. In the first case here, we are deciding not on the alcohol, but we are designing on the tail concept here. Okay. So when you look about tail, they are completely hydrophobic. So where they are water
Phobic.
hating. But in first case, the length of the fatty acid is very small. In second case, the
Mhm.
length of fatty acid is very high. Now the question is, which of them can show you rapid transverse exchange, that is lateral diffusion, fast movement. Which of them can give me the fast
Mm.
movement? So based on the diagram, we can understand that who is having the fastest movement? The lipids which are small in size or
The
lipids
small.
which are larger in size?
Mhm.
Lipids which are small in size, they have a faster movement. Okay, beta.
Yes, sir.
Now a simple thing I'll ask you. You take one chili and you just bite. எப்படி
Mhm. Mm.
எப்படி இருக்கும்? ஒரு சும்மா ஒரு மிளகா எடுத்துக்கோங்க. ஒரே ஒரு கடி கடி எப்படி இருக்கும்?
Mm.
காரமா இருக்குமா இல்லையா?
Sir.
Now, beta, any voice lagging? Can you hear me?
Yes, sir.
Yeah. Second thing, I'm going to take the same the lipid molecule. So one is small in size, and other one is we are going to take in large in size. So smaller the lipid, faster the movement, faster the fluidity. Are you able to understand this?
Yes, sir.
Now, second, I told you, you have two types of oil. One is coconut oil. When you keep in the refrigerator, they will get solidified. Or the same time,
Mhm.
when we keep this refined oil or olive oil or castor oil, when you keep in refrigerator, they will not solidify. We call them as unsaturated fatty acids, right?
Yes, sir.
So unsaturated means what? In unsaturated fatty acid, there is always a double bond. Saturated are, they have a double or triple bond. Saturated, they have only single bond. They don't have double bond. So double or triple bond makes them stable. At the same time, unsaturated are components are highly stable compared to saturated. So in
Mm.
unsaturated compounds, the rate of transport of molecules are faster. Okay.
Yes, sir.
Third thing is cholesterol content. So you have a cholesterol content here. So we look about the
Mm.
cholesterol content, one which have a higher number of cholesterol also, the rate of movement is faster. Temperature, when the temperature increases beyond a certain limit, the plasma membrane proteins as well as lipids will get denatured. So the rate of
Yes, sir.
transport of molecules increases with increase in temperature. I just have a graph to understand this, x-axis and the y-axis.
So, when we are learning this, sir, you're saying the faster the movement. So, what is the movement you're mentioning here, sir? In this
Movement of
case, it is
molecules
the
inside,
transfer section.
now that is inside and outside the molecules of the plasma membrane, that is between the cell and the outer medium. How is the speed of molecule, the rate of diffusion or rate of exchange of gases, how they communicate and how they exchange the molecules? It is a tough one or a easy one, that's what we are discussing. Okay, better.
Yes, sir. So, like the the molecules that are responsible. So, like in the plasma membrane, so we learnt about the integral proteins, no, sir.
Hmm.
So, the movement of the proteins that are necessary, molecules that are necessary for the cell, they're talking about in this case, sir.
Hmm. Yes, beta.
Okay.
Now, yes,
So,
yes,
sorry,
yes,
sir, sorry, sir, one more.
yes.
The molecules that are responsible for entering and also also the molecules that are surrounding around the plasma membrane also is
Yeah, that is,
included in this
yeah,
case,
both
sir.
entry and exit of molecules. Both I'm discussing. Entry from outside to inside the
Okay.
cell, exit from inside the cell to
Mm-hmm.
outside the cell. Both I'm discussing.
Okay. Okay,
X
sir.
axis, Y axis. So when I look about the membrane activity, so in the Y axis, I'm just checking into consideration the membrane activity. That is
Mm-hmm.
the lateral transport of molecules. In the x axis, let me go with temperature. At lower temperature, membrane activity is low. When we keep on increase
Mm.
the temperature activity increases. After certain limit, it's not at all increasing, then it will keep on decrease. Got it?
Yes, sir.
Why there is a decrease means this temperature where you can see this maximum activity, this one is called as
Mm-hmm.
your optimal temperature. Higher temperature, what will occur? The enzymes, because this occurs only by enzyme, right? Flipases.
Yes, sir.
So
Yes,
at
sir.
higher temperature, enzymes are getting denatured. For example, making an omelette, add a
Denatured.
protein, they will lose their
Mm.
activity, right? They will become solid in nature. So higher temperature,
Yes, sir.
enzymes will get denatured. That is the main reason that why in temperature the rate of activity of or the rate of transport of molecules to the plasma membrane is slow and another area it is faster. Are you able to understand this?
Sir, can you mention it again, sir? I could not hear you.
Which one? Which one? Which one? Which one?
Like the I have a doubt in cholesterol part, sir.
Hmm.
The last line I couldn't understand, sir.
Okay. The first thing, are you able to understand about the length of the hydrocarbon?
Yes, sir.
Okay, that no problem, right? You're able
No
to
problem,
understand
sir.
the based on the number of bonds, the saturated and unsaturated acid. Okay, before that, which one is very
Yes,
good
sir.
for your health, ma? Saturated fatty acid or unsaturated fatty acids?
Can you repeat it, sir?
Which one is very good for your health? Saturated or unsaturated fatty acid?
I guess it is saturated.
Sure. Saturated are solid at room temperature, right? Like coconut oil.
Oh.
So
Okay, so unsaturated.
if I prepare your food only with butter, only with cheese, or
Um,
only with palm oil,
um,
It's good for your health.
No, sir.
Then which is very good for your health?
Saturated.
Yes. So when you look about
Okay.
here, saturated is good or unsaturated? Saturated, listen here, beta. Saturated is like coconut oil. They are solid at room
Okay.
temperature. So what is blood clot, or you can say what is CAD? Coronary artery disease. You have a blood arteries are there. There you can get cholesterol accumulation, right?
Yes, sir.
So this will give a
Yes, sir.
block. So blood flow will become minimum, right? So if I take
Yes, sir.
any oil items, and if the oil items are saturated, then at the given temperature they'll become a solid, right? So it's bad for your health.
Yes, sir.
At the
Yeah.
same time, unsaturated are liquid at room temperature. Even if you keep in the refrigerator and all, you just try puja. You just take some cooking oil and keep
Yes.
in the deep refrigerator in your home. After
Mm.
1 hour, you check, it will not get solidified easily.
Mm.
It will be more or less semi
Okay, sir.
liquid or liquid in nature. Pure means it will become as complete liquid. It's a somewhat average item means it will become semi solid. At the same time, coconut oil, if you keep and see, it will become solid immediately.
Yes, sir.
So, which one is very good for your health means unsaturated because they are liquid at room temperature.
Saturated.
So, unsaturated will increase the rate of membrane activity.
Okay, sir.
Next is temperature. Are you able to understand?
Uh, yes, sir. Temperature was able to understand,
Now,
sir.
I'll go with cholesterol. Okay. Since you had a doubt with cholesterol, I'll go with cholesterol. Okay. Now, what is actually cholesterol in case of plasma membrane? Look about
It belongs
cholesterol.
to sterol.
Okay, it's a
Uh,
type of steroids. It's fine. It's what will actually, it is a stabilizer agent.
yes, sir.
Commonly, it is a stabilizer agent. So only when you ask your doubt, one day I'll be going in depth. Okay.
Okay, sir.
So what happens here means this cholesterol, why they call stabilizer means they fill the gap. They fill the gap between. You can see in this diagram. Do they have small gaps here?
Yes, sir.
You can see some gaps are there here now. Between the spherical structure,
Yes,
can you
sir.
see some gaps which are given in black in color?
Yes, sir.
So they'll fill the gap which is made between lipids, especially phospholipid, a phosphorus containing conjugate lipid. Phospholipid.
Mm.
So they go and fill the gap which is there between, which is between the phospholipid. So when the gaps are
Okay.
filled up, so what happen is when you heat it to the higher temperature, so it will become less fluid and more strong. As a result of that, what occurs generally the flow of, or you can sense the so becomes so hard, and when cholesterol is actually they go and fill the gap. So when they go and fill the gaps, I'll just zoom the diagram. Okay, I'll just go with the diagram here itself. So imagine if you have a cholesterol molecules here. This is
Mhm. Glycerol.
your glycerol, and this one is your fatty acids. So if cholesterol concentration increase, let me give in yellow in color. Yellow is visible. Yeah. If I give yellow color here, if cholesterol is covering these joints, then the dynamic
Mhm.
nature of the membrane will be lost, right? See,
Sir.
when I cover this with fat layer, especially see, I'm covering
Huh?
this not with the gaps. When I'm covering this with fat layer, that is cholesterol layer.
Yes, sir.
Now tell me, is the
Uh-huh.
hydrophilic end exposed?
No, sir.
At the same time, if I fill only these gaps, If only these gaps alone are filled,
Uh-huh.
then what happens? There's partial exposure of hydrophilic end and complete inhibition of hydrophobic end have taken place.
Yes, sir.
Here, in this case, it is complete inhibition of uh, amphiphilic end. Am I right?
Yes, sir.
But here it is partial inhibition of hydro both them of hydrophilic and complete inhibition of
Hydrophobic.
hydrophobic end. Since they become a completely, they have become stopped here. Now tell me, will the flow of molecules will be faster or slower?
In the second case, partial inhibition, it will be faster, sir.
So this is what I'm going to. So here it will be faster, it will be slower.
Ah,
Therefore, increase
yes, sir.
in cholesterol
Hmm.
will decrease the membrane activity. Do you agree with this?
Yes, sir.
Because they are filling the gaps and they are covering the entire
The visceral part also.
yeah, that is they covering the flowing part of your membranes are completely covered. Because of this, the movement of molecules are highly limited. Are you able to understand now?
Yes, sir.
Any doubt? See, Pooja,
No, sir.
first you should
Sir,
understand.
one.
Yes, Miss Ma.
Yes, sir. Sir, in this case, you're talking about enzymes, no, sir. Mostly the enzyme they're talking about in this case is flipases, no, sir.
Lipases will destroy the lipid, ma, not lipases, ma.
No,
No,
flipases, sir.
lipases.
Like this one.
Hmm.
Ah,
Yes, beta. I'm talking
okay,
about the
sir.
enzyme called flipases.
Yes, yes, sir.
So this flipases enzymes will get destroyed because it's actually an enzyme.
Yes, sir.
This enzyme at higher temperature they get denatured, they get destroyed. That is
Yes,
why
sir.
the membrane activity has been decreased when there is increase in temperature. So when this increase in
Yes, sir.
temperature, there is decrease in membrane activity. Got it?
Yes, sir.
So, see, Pooja, why you are dealing with this is even next topic is all about sphingomyelin and all. You're going to learn about sphingomyelin, cholesterol, how do they
Okay, sir.
organize? All this we are going to discuss.
Uh,
We have some background voice.
Sir?
Some background voice. Okay,
Yes, sir. My father
That's
speaking,
fine. That's
sir.
fine.
Sorry, sir.
That's fine. No issues. No issues. So what happens here
Okay.
when these membranes are
അവിടെ
exposed
കാശ്
to
കൊടുത്തല്ലേ
different
പെട്ടിയാളാ
environment? That is, when a cell, we take about a cell, the cells are not only here, they're exposed to different types of environment. So when they're exposed to different types of environment, definitely they are going to get destroyed so soon. Because plasma membrane should be always active. Only when plasma membrane is active, that is inflow of chemicals or outflow of chemicals or inflow and outflow of molecules can take place. The signaling molecule can be transported. The signaling chemicals can be reported. For example, if I take a simple one called as your hypothalamus or your pituitary or your thymus, thymus is also made up of thyminic cells. Your stomach is made up of oxytic cells. So when all these have to function, mean plasma membrane should be always active. So for the dynamic nature of plasma membrane, which one will take place? Which is responsible for the dynamic nature of plasma membrane, Pooja?
Cholesterol,
Pooja?
sir.
Yeah, it's which one?
The cholesterol is responsible for making the plasma membrane dynamic, no, sir?
Oh my god, no, no, no. What cholesterol? Lipids. Hey, this is why I discussed here.
Yes, sir.
Ma, the
So
fluidity
for
is because
fluidity,
fluidity is because of lipids. So fluidity means the speed of
yes,
action
sir.
of the membrane, right?
I understood, sir, but like in the previous class, you said that for plasma membrane to be dynamic, we also need cholesterol, sir.
Yeah, we require cholesterol because without cholesterol, the plasma membrane cannot function. But all required in optimum level, required level. The point I've given is
Sir,
increase in cholesterol above the normal will decrease the membrane activity.
yes, sir.
So my question is, which part of the plasma membrane is responsible
Uh-huh.
for the dynamic nature or membrane fluidity? Option A,
Sorry, sir.
protein. Option B,
Lipids.
lipids.
Okay. Lipids, sir.
Lipids only are responsible for their fluidity. Fluidity means the dynamic nature. The plasma membrane is always active. So, plasma membrane to be always
Sir.
active only a cell is functioning. If plasma membrane is not at all functioning, means the exchange of chemicals cannot take place. Your cell cannot give you any secretions. You have a pancreatic cell. Example is uh, beta cells of Langerhans. What do they give you? Insulin.
Yes, sir.
Yeah, pancreas will give you the insulin, right?
Yes, sir.
So pancreas is made up of pancreatic cell.
Uh,
So pancreatic cell membrane should be functioning so that they can deliver the insulin outside the cell, right?
Yes, sir.
If the plasma membrane is not functioning, if there's no lipid molecule means how will be the membrane exist or fully covered by cholesterol also? Cholesterol will stabilize the plasma membrane. But above the normal it is there means how the secretion can cross the cell and they come out, they cannot come out, right? See this diagram. How the
Yes,
secretion
sir.
can come in and out of the cell, it's not possible at all.
Yes, sir.
So today what we have learned is the fluidity of the plasma membrane is because of not proteins but because of lipids. This fluidity
Lipids.
says that plasma membrane is dynamic. That is a small experiment by which we can understand that lower temperature no changes, higher temperature changes. Why lower temperature no changes? Because lipids will be losing their enzyme activity at a lower temperature. Okay,
Yes, sir.
and plasma membrane only allows the lateral transfer of molecules by the enzyme called flipases. This flipases under a higher temperature they get denatured, so enzyme activity is lowered, so fluidity memory of the property is also lost. Cholesterol is required to the required level, but remember this point. Cholesterol, if you want, if making a note of cholesterol somewhere, that is cholesterol will increase the activity. You make the note. Cholesterol content above the normal will fill the gap
Uh-huh.
between the phospholipid, which, which may lead to complete inhibition of amphibilic end or partial inhibition of amphibilic end. As a result, the fluidity of membrane may be lost. If the concentration of cholesterol is increasing, the number of double bonds or more means they are liquid at room temperature because of unsaturated molecules generally they have double or triple bonds. Alkanes and alkenes and alkynes படிச்சிருக்கியாடாமா?
Yes, sir.
அதாம்மா. So unsaturated compounds they have double or triple bonds. So because of that they are liquid at room temperature. Plasma membrane are lipids, so they should be liquid at room temperature. So more the unsaturated compounds, faster the activity, and smaller the fatty
Yes,
acids,
sir.
faster the activity. This is a very important
Yes,
concept. Are
sir.
you able to understand?
Yes, sir.
Pooja, slides are only two in number. See, I've discussed only this one, two. This is not three. Three slides, but it took 1 hour to clear your concept. Are you able to understand? So, but it is a very important thing because
Yes, sir.
in your syllabus you'll be doing a lot of research. For that, these are a very
Yes.
important basics. Okay.
Okay,
So
sir.
next is
Sir, I have one more doubt, sir.
yes,
In
ma'am.
this fluid mosaic model, sir,
Hmm.
here we are talking about only the movement of molecules. through the passage of integral proteins that are needed for the cell.
Hmm.
Yes, sir.
Exactly. Where's the diagram here? Yes, we are talking about the channel proteins.
Yes, sir.
So the channel protein should be free. Imagine this. Imagine that if the lipid molecules are so tight, they're not able to adjust with this. Then how the membrane will move? Only when the
Sir.
plasma membrane, the lipid layer are free enough and the liquid nature only, these membranes can do their activity. When they store stubborn, then the door cannot be opened because the channel protein has a door. That's called as selective channel pore. That will not get opened.
Yes, sir.
So they have to be dynamic. When everything is solidified, proteins cannot do their activity because the liquid part should be this bilipid layer. The bilipid layer are not active means it will become so rigid and strong like a wood. Then how the molecules can pass through? It's not possible, right?
Yes, sir.
Wait, I'll stop the recording first and